yes, today I'm here to talk about viruses and their k-mer preferences. And really, we think this is important, that there really is a k-mer preference and not just some arbitrary sort of average of k-mer spectrum here. So to introduce this, I'll talk a little bit about virus genomes. And they're composed of either DNA or RNA. Both single-stranded and double-stranded variants of this exists, and they're quite remarkable due to the fact that there's so huge variation between the lengths of viruses. So, for instance, one of the first, or I think the first virus discovered is this 6,000 nucleotides long genome consisting of RNA. And the RNA viruses grow to be around 30 or 40,000 nucleotides, and coronaviruses are one of the larger RNA viruses then. But on the DNA side, these viruses can actually, for instance, the human herpes virus is 152,000 nucleotides, and the largest today known virus is 2.5 million nucleotides. So there's a huge variation in both what types of genomes they use and how they're how they work. And in comparison to other classes, we can see that RNA viruses, as I mentioned, they're pretty short. DNA viruses actually rival bacteria in terms of genome size. Of course, the number of DNA viruses that are this large are quite small, but still it's quite remarkable. And even some eukaryotes are sort of similar in genome size. But this short genome also means that a lot of their genomes are coding, which is not true for a lot of these eukaryotic genomes, where we have a lot of non-coding and other stuff that goes on in the genome that isn't necessarily produced into proteins. So one of the sort of key things about viruses is that they're completely dependent on their hosts for replication. And this is just one example of a virus gaining entry into a host cell, where then the host's cellular machinery are sort of responsible for producing protein, and in some cases, especially for some DNA viruses, also replicating the genome. And we think that this is important and on the genome level, because there are a lot of indications that some specific genomic features are important for the host in their sort of host's cellular environment. For instance, nucleotide content might be important. This is just one example where we have a GC content of 0.4, for instance. Dinucleotides have also been shown to have some sort of importance for both for viruses for evading hosts' immune responses, but also perhaps for other reasons. And this has been shown to be very specific across a lot of different organisms, the dinucleotide usage. And so on, codon usage can also be important. And for instance, we might see that some proteins that are highly expressed have a certain preference for some types of codons. And clearly, even in this gene, there's quite a large difference between, I've included the most common codon and the least common codons here, including the stop codon, of course. And that's true also, you see dinucleotides come in very different frequencies, and also codon pairs, which we also think might be relevant for translational stability and other things. So to study this sort of impact of not just how viruses use code or use their genomics features, but also what type of impact the genomic features have, or the cellular environment of the virus, of the host of the virus has on the genome, we're not necessarily want to look only on these features, but instead, of course, we look at k-mers in general. And again, we see this sort of huge difference between different frequencies of codons, or of k-mers rather, and of course k-mers, k being 1 mers, 2 mers, etc. But due to the fact that we see this huge variety in sequence lengths across the viral spectrum, we don't want to look at absolute frequencies of k-mers because that would be sort of hard to reason about, but instead we're looking at the probability of observing specific nucleotides in the genome and dependent frequencies or probabilities of observing, for instance, a T after having seen an A previously in the genome, which comes from the dinucleotide AT, of course. And we can extend this sort of context. So we can look at probability of observing a T after an A, or observing a T after AT, and so on. And this becomes quickly very large. And we have a lot of parameters here. Growing this context means that we include a lot of things, and not all of those things will have very statistical significance, because we see that, for instance, this is sorry, instead of using that, we're using a variable length Markov chain. And what this does is it allows us to not use all of those parameters, but only the parameters that we think are important in the genome. And how we characterize that is by looking at in which cases are the differences when we add this sort of context, in which cases does that matter? So in this simple example, we have the probabilities of observing each nucleotide after an A, for instance, and after CA. But perhaps the reason for not including AA here is that those probabilities are very similar to what we already observed for the just the context of knowing that there was an A previously in the sequence. And that allows us to remove a lot of those unnecessary parameters in the model, and allows us to make hopefully a better model of the genome. Which we also recently have developed this new visualization for. So each sort of chart or area here represents the probability of observing each sort of k-mer, but of course not all k-mers are present because they're missing in the variable Markov chain. So in order to look at this, we want to discover how specific is the usage of k-mers for each virus. And as I mentioned, we don't want to just know that the average k-mer frequency is unique for each virus. That's not very important. Instead, what we want to know is that sort of relative frequency of each k-mer reoccurs throughout the genome. So to do that, we split each virus sequence into two parts. We call one part the query, in this case, and another part the signature. And for each signature part, we compute our variable Markov chain. And that allows us to then score each query based on what has the highest likelihood of sort of coming from the same sequence originally. And then we look at, are those viruses, or are those two from the same species? Are they from the same family? Or what are the other types of relation we can see here? So what we look at is sensitivity of, which is the fraction of queries with the same species, for instance, in the top 1. So if the very most likely signature and query come from the same species, in the top 5, if for a query we have the correct species somewhere among the 5 most likely models, and so on. And what we see is that for very long viruses, this is almost always the case, that they have a conserved preference for some type of k-mer within their genomes. But on the other hand, for very short viruses, this doesn't seem to be the case. But still, it's not insignificant. We compare this in all cases to a random model where we just randomly assign virus query and signature parts. And in all cases, that is much worse than the actual signal. So if we instead look at the genus and the family of viruses, we see that if we look at the top 5 hits, in all cases, in more than 50% of cases, we have the same genus, and for both of the query and the signature. And for the family, we have even more, above 70% of cases that's true. So what this means is that, or at least how we interpret this, is that there does seem to be some preference for k-mers within these viral genomes. Although for short viruses, this might be not either difficult to detect because they are so short, and remember, we're still cutting them up even to be even shorter. Or it might mean that longer viruses, they have more genes, so maybe they have some other sort of stability that they that makes it possible to sort of preserve this k-mer preference. If we instead look at viruses by their Baltimore classification, which is really their type of genome, so double-stranded DNA or single-stranded RNA, for instance, you see that double-stranded DNA, among all the DNA viruses, they have this preference for some k-mers much more prominently than single-stranded DNA do, which is not the case for RNA viruses, where we see sort of an equal scores across all of this double-stranded and single-stranded versions, but the retrotranscribing, so the retroviruses, have much higher scores in general. And diving even further into sort of the different families, we see that even here, there's a huge variety between different families within even the double-stranded DNA, where we saw the highest level of conservation or preference. For instance, the adenoviruses have, in some cases, no similarities. So, and this is only looking at the very most similar here, so not including the 5 most similar signatures. While on the other hand, so for instance, herpes viruses have a much stronger preference for some specific k-mer usage. And similarly, among the single-stranded RNA viruses, we also see this variety where, for instance, coronaviruses, which I mentioned are the longer, one of the longest groups of viruses, RNA viruses have the highest, or one of the highest degrees of this sort of preference, while some other RNA viruses are not. This doesn't seem to be as important. And using this, we can also, these sort of similarities between the query and signatures, we can also build a neighbor joining tree of all of these viruses that we include to explore not just are all of the viruses in, or are a virus similar to any signature or query similar to any signature, but is this really conserved within the entire group, which doesn't necessarily seem to be the case. For instance, we saw previously that herpes viruses have a high preference for sort of having a very similar or having a strong k-mer preference, but we also see that a lot of different herpes viruses are placed sort of not randomly, but throughout this tree, which indicates to us that perhaps there's a huge variety within the herpes virus genomes, when there might be preference for different things in different sort of areas of that group. And on the other hand, all of the coronaviruses are all really similar up here. And that's true for some other groups as well, but still. So um, so I hope I've convinced you that at least some viruses have a strong preference for adapting towards a specific k-mer preference. But what does that say in terms of how similar, I mean, how does that cellular environment of the host impact this? And to do that, we have to also look at the genomes of the hosts, right? And just to remind you, viruses rely on their host to produce their protein, yeah, or at least in many cases. So that that should have an impact on the genome. At least that's the sort of idea. But in order to do this, because, and I mentioned that viruses have much shorter genomes than some of the eukaryotes which they infect. So in order to do this, we've also had to develop a couple of new algorithms to actually construct our variable length markup chains on these much larger genomes. And what was previously not, so this is a previous algorithm for this task. And we've developed two new algorithms, and the fastest, our fastest version of this even works on the largest sequenced genomes to date in a sort of a reasonable amount of time, which means that we can now also analyze similarities in this variable length markup chains between the virus and its host genome. Although we do only look at the coding regions of the host genomes. So they're not, the difference isn't that large as it would have been if we were looking at the entire genomes. So what we do in this case is that we compute the genomic features for every sequence, and we've been a bit more careful here in that we want to look at all of the different features, not just the aggregate of features. So we'll see later on, we have a lot of, so we're looking at GC individually compared to dinucleotides and so on. And then we compare these features directly. And then we're looking at, are viruses similar to their hosts? Are viruses with the same host? Are they more similar than would be expected from their sort of phylogenetic relationship and so on? So this is one example of a bacteriophage, so a virus that infects bacteria, and its similarity to hosts of the correct species that it actually infects, and then more distantly related hosts to that. So, for instance, members of the same phylum as the host. And we can see here that the similarity, and I see that the y-axis has sort of disappeared here, but never mind. We see that the GC content similarity, at least, is much more similar for the correct host species, while the host phylum is not as similar. So clearly, we're seeing here that these viruses have a similar GC content to their hosts, but when we correct all of the other, so 2-mers, 3-mers, 4-mers, etc., for the GC content, this doesn't seem to impact that as much. But when we do this analysis, we also have to bear in mind that a lot of these hosts are also very similar to each other. So for instance, if we look at the dinucleotide usage, if we correct that for 1-mers or for GC content, all of the eukaryotic families are already very similar. So if we see that a virus is similar to its host, it's also likely similar to other hosts, which might decrease the sort of signal that we see here. And also, notably, the variation in these values is much larger for viruses, which I think we know, but it's important to keep in mind for this sort of analysis. So the next step is then to look at how similar are viruses from the same genus that infect the same hosts. And then we compare that to the entire similarity in the entire genus. and then similarly for the families. And we're looking for a slope here, and the slope means that they're more similar to the similarity within the group of viruses that infect the same host is larger than the entire similarity in the entire family. And these are some of the most interesting and some of the most prominent cases where we actually see some sort of adaptation or similarity here. But of course, this doesn't account for how similar a lot of these viruses may be, even if they are in the same genus or if they are in the same family, which is something that we're looking into in the future to sort of correct for that. But nonetheless, here it does appear that some viruses, at least, but I should say that this is only 6 families out of maybe 150 that we include, or yeah. So a lot of viruses don't have this similarity to their hosts, and also don't have similarities to other viruses with the same host. So what does that say about the Kamer preference? If it's not caused by the sort of an adaptation towards something that we can measure in terms of similar adaptation of the codon usage, for instance, that likely means that this is some pressure put on the virus that's evolutionary favorable for some other reason. And what those reasons are, we're not entirely sure at this point. Some ideas are that perhaps this makes it more, perhaps this makes it so that viruses can recombine more efficiently, which increases their stability and reduces deleterious mutations, and so on. Or perhaps there are some other reasons that biological reasons that we don't know about yet. But nonetheless, we hope that this idea of studying these Kamer preferences in viruses allows us to eventually produce an algorithm that can detect viruses in metagenomic samples, even if we don't have a clear reference genome, or if we don't have a strong homology to existing viruses already. Thanks for listening.